Hello and welcome everyone to my channel, the MBBS Hub. Today our topic is the histology of spleen. So let's start our today's topic. So the spleen is a hemolymph organ of the body and it is one of the largest lymphoid organ. Now the major function of spleen is blood formation in the fetal life and blood destroying organ in the postnatal life. It filters the blood and it's an important organ in the defense in blood-borne antigens and microorganisms. Now, if we macroscopically cut a section of spleen, then it bears numerous discrete white nodules which are also known as the white pulp and some aggregations of the lymphoid tissue in the red matrix called the red pulp. So, we can see small discrete white nodules which are called the white pulp and some aggregations of lymphoid tissue in the red matrix which are known as the red pulp. So this is the structure of the spleen and it is present in our human body just in the left side to the stomach. Now structure. The surface of the spleen is covered by a layer of peritoneum, that is the serous coat, except at the hilum. Except the hilum, the total surface of the spleen is covered by a layer of peritoneum. Now deep to the serous layer, the organ is covered completely by a fibroelastic coat which is called the capsule. Now when we go deep to the serous layer, then we get a covering of fibroelastic coat which is called the capsule, and there are some trabeculae that arise from the capsule and extend into the substance of the spleen. Now trabeculae divide into smaller branches and form a network. So the parenchyma, the interior of the spleen shows round splenic pulps. Now there are two kinds of pulps or two types of pulps. Number one is the white areas or the white pulps, and the white pulps are surrounded by the red matrix which is also known as the red pulp. So we will learn about white pulp and red pulp in details. White pulp. Now white pulp is made up of aggregations of lymphocytes that surround the arterioles. In the HN stain, the white pulp appears basophilic because of dense heterochromatin of the lymphocytes. Within the white pulp, the arteriole called the central arteriole is present, and the position of the arteriole is eccentric in position, that is, it is not in the center of the white pulp, it is a bit away from the center. The lymphatic aggregation around the central arteriole constitute the periarterial lymphatic sheath or the PLS. On antigenic stimulation, the B cells present in the middle, means there are some B cells which are present in the middle. These B cells proliferate and they form the germinal center. This pushes the arteriole to periphery. So what pushes the artery, or what is responsible for the eccentric position of the central arteriole? Now this is the B cells which proliferate and form the germinal center, and that pushes the arteriole to the periphery. The functional significance of the white pulp is like that of cortical tissue of lymph nodes. The germinal centers are the areas where the B lymphocytes are dividing. So now we will study about the red pulp. Now macroscopically the red pulp appears red, and even in the histological sections because it contains large number of red blood cells. Now what is the reason of the red pulp appearing red? The reason is the red blood cells. Splenic cords. 75% of the splenic parenchyma is formed by red pulp that contains splenic cords, and these are also known as cords of Billroth and sinusoids. Now splenic sinuses. Splenic sinusoids separate the splenic cords, and it is lined by unusual elongated endothelial cells. That is, the splenic sinuses separate the splenic cords and these are lined by unusual elongated endothelial cells, and these cells are known as stave cells. This is the diagram of spleen. So this is the capsule. After that, these are the trabeculae. This is the germinal center which is formed by the proliferation of the B cells or B lymphocyte cells. This is the white pulp. After that we can see the eccentric position of central arteriole in eccentric position. Then we will see the red pulp which is surrounding the white pulp. Then this is a blood vessel. Now we are moving towards the microscopic view of the spleen. Now in the microscopic view of the spleen, first here we can see an eccentric central arteriole. The position is eccentric of the central arteriole. After that, this is the white pulp, and this is the germinal center. Next is the blood vessel. This is small blood vessel, which can be seen here. After that, the red pulp, and this is the trabeculae that is present in the spleen. Now we will move towards the identification points. Spleen is characterized by a thick capsule with trabeculae extending from it to the organ. Trabeculae is extending from the thick capsule to the substance of the organ. Now we will see the white pulp and red pulp when we will see the slide of spleen. The white pulp has nodules with germinal center and periphery placed central arteriole. Now what are the features of the white pulp? The white pulps have nodules with germinal center, and they will have a central arteriole which will be placed peripherally or the eccentric position. Now red pulp is diffusely distributed lymphocytes and numerous cords. Okay, so the main thing which we have to identify in a slide for spleen is the red pulp and the white pulp. Now how can we identify white pulp? White pulp will have nodules with the germinal central and will have a peripherally placed central arteriole, and the red pulp will be distributed diffusely. With this we are ending our today's lecture. Hope you people have find the video informative. Please like the video, subscribe the channel and share the video among your friends. See you in next video. Thank you all.